Hello, my name is Saida Gamidova. I am an undergraduate in Dr. Paul Katz's lab at UMass Amherst. I work closely with Otter, who is a PhD candidate in the lab, and today I'm going to be sharing with you the behavioral basis of group feeding by the nudibranch mollusk Berghia stephanieae. So there are many animals that engage in social feeding. You have humans, of course, lions, flamingos, land ants, just to name a few. So there are, of course, benefits and drawbacks to group feeding. Some of the benefits include exert less energy, you can spend less time hunting, you're capable of finding more prey because there are more eyes and ears looking out for it. And then the one that pertains to our lab is that you sustain fewer injuries. And then drawbacks include you have to interact with conspecifics, it may be easier to be spotted by predators if you're in a larger group, and then you're competing for resources and prey. So I'd just like to reiterate that feeding in groups can help animals consume larger, more dangerous prey, as shown in this image to the right of multiple wolves taking down a large caribou. So the Berghia stephanieae feeds on the sea anemone known as Exaiptasia pallida, which is a dangerous prey item. And from this video you can see a large sea anemone with the Berghia at its center, and this Berghia is trying to break free from the anemone's grasp because it is being injured from the nematocysts, which I will explain in the next slide. So what makes the sea anemones dangerous is that they have these stinging nematocysts, which this image is pointing to right here. Here are the tentacles, and the nematocysts are found on those tentacles. This, of course, brings me to my question. So do Berghia use group feeding as a strategy? When we observe these Berghia feeding in the lab, they tend to feed in these large mobs or groups. So the image on the left shows 8 Berghia feeding around one anemone, and then 5 minutes later you have this image on the right which shows 12 Berghia feeding on that same anemone. So they do exhibit group feeding in the lab. So after observing the Berghia feeding in the lab, we saw that they didn't distribute evenly amongst the anemones. And then from the image on the left, you can see one anemone with 0 Berghia surrounding it, and then you have another with 10 Berghia surrounding it. And then at the bottom, you have one anemone with 5 Berghia on it, and another with 4. And so this graph on the right was put together collecting the data from 21 tanks, and what we found is that most of the tanks were above this 50%, so that means that more Berghia were likely to be on one anemone over another rather than evenly split. So what we wanted to find out is, do Berghia feed socially if given the option to feed alone? So what we did is we placed 8 anemones in a circle around 8 Berghia that were placed in the center of the tank. And then this video just shows one of the trials that we had, and they ran for 20 minutes. And as you can see in the bottom anemone, you have a group of 4 Berghia feeding around one anemone while the other ones are dispersed amongst the other ones. So one thing that I like to point out is that all the anemones are equidistant from one another in the center, so there's not one that's closer than the other. And what we did is we took our data and compared it to simulated data, and our simulated null hypothesis, which was that each animal would choose independently with equal probability going to one anemone over another. So what these graphs show is that our behavioral data in the box plot at the bottom, which is our observed mean group sizes, they were larger than the simulation had predicted. So this means that the Berghia aren't choosing independently of each other, and that they're forming larger groups than what was expected. So do Berghia use group feeding as a strategy? And what we found is yes, but what we wanted to know next is what mechanisms they're using. The next thing we decided to look into is the mechanisms that the Berghia may be using to form these groups when feeding. The first mechanism we looked at is slime trails. So Berghia leave a trail of slime when they're in motion or stationary, and that could be detected and followed by other conspecifics. Second mechanism we looked into is injured anemones and odorants. So Berghia may be able to sense when an Exaiptasia is injured, and this may make it easier to find and prey on. And the last mechanism being feeding pheromones. So Berghia may be secreting a pheromone when feeding that other Berghia could be detecting and following. The first mechanism I'm going to discuss is the Berghia following conspecific slime trails that can lead up to the prey. So this video shows a helper slug creating the slime trail, and it is going to the anemone, and then you have the focal slug, which encounters the slime trail and then goes to the same anemone as the helper slug. So from our results, we can show that Berghia have no preference for an anemone with a slime trail leading to it. And as you can see from this graph, you have this error bars, which represents the 95% confidence interval, and this 0.5 red line, which is 50-50 chance. And since our data falls within that red line, that 50-50 chance, it indicates that there is no preference for slime trails versus not. We decided to analyze the paths of the Berghia and their slime trails, and some of the results that we found were a little strange. Sometimes the focal slug doesn't come in contact with the slime trail that was created by the helper slug. In the video on the left that just played, it only came in contact at the last minute. And in the video on the right, the helper slug creates the slime trail, but then the focal slug comes in contact with that slime trail and decides to feed on the other anemone, which is not something that we expect to see. So the next mechanism we looked at is, do Berghia prefer the odor of an injured anemone versus one that's intact? So the first thing we did is we cut the anemones in half, and then we used just fresh artificial seawater. And what the graph on the right shows is this 95% confidence interval falling within a 0.5 red line, which shows that there was no preference for an injured anemone versus an intact one. So continuing on with mechanism 2, we did the exact same experiment, but this time we used anemones that had been eaten rather than cut, and we also put the Berghia in anemone treated water rather than fresh artificial seawater. And what we found is that they showed a slight preference for intact anemones versus over injured anemones. The third mechanism we looked at, of course, is Berghia may be attracted to the scent of an anemone that is being fed on by a conspecific rather than one that was intact. So again, our results weren't what we had expected. The Berghia seemed to show preference for the intact anemone and just the fresh artificial seawater, but then when we did the exact same experiment in anemone treated water, there seemed to be no preference. So just to summarize our results, we didn't get any of the results that we predicted to get. As you can see, for many of the mechanisms, we either got the opposite of what was predicted or there was no significance. So overall, our findings are contradictory. We found that Berghia feed in groups, even if they are given the option to feed alone. But then we tried to look into how these Berghia may be formed in groups. We found that they don't appear to follow slime trails. They prefer uninjured anemones over those that are injured. And then they also prefer anemones without a conspecific feeding on it, unless there are olfactory cues, which show no preference. So some questions that this leaves us with is, what if we used a combination of some of the cues, like slime trails with an injured anemone or slime trails with an anemone already being fed on? And then we also want to know if there's a threshold. So if we used 3 Berghia instead of 2, we can find different results. And then there also could be this trade-off between sustaining less injuries and getting more food. So we could do the same trials with 3-day or 5-day food-deprived Berghia to see if that changes anything. So I'd just like to thank the Katz lab, Professor Paul Katz, Kate Otter, UMass Amherst's Brain Initiative Project, LISIP, and UMass Stats Consultants. And thank you all for your attention, and please reach out to me or Kate Otter if you have any questions or ideas for us. Thank you.